Let's imagine that we shrink down and we get into a little ship and we're going to approach a cell from the outside. What would we see first? Well, that would really depend on what kind of cell it is that we are going to explore. If it's a plant cell, the first thing that we are going to encounter is the cell wall. The cell wall is a rigid outer coating made primarily out of cellulose. There's some other materials in there as well. There's lignin and these kind of things. But the thing you're going to want to remember is that it's primarily made out of cellulose. Now, you may remember cellulose from Unit 1. I hope you do. It is a polysaccharide made out of glucose subunits. Glucose is a monomer, cellulose is the polymer, and it has these strings of glucose put together in such a way that every other glucose is turned upside down, which allows hydrogen bonding in between these strands of cellulose, and that creates these fibrils, which are very, very strong. This allows a lot of structural support. So the plant cell walls are made out of cellulose, and this helps protect cells and maintain their shape, and it also prevents the cells from absorbing too much water, which I alluded to in the previous video. Now, cell walls are not completely impenetrable. You need to be able to let stuff in and out of your cell, so they have holes in them that allow passage of materials from one cell to another cell. These holes, these portals, are called plasmodesmata, and this allows water and small solutes, sometimes things like protein and RNA, to pass from one cell to another cell. So you got your cell wall, nice rigid support, but maybe it's not a plant cell that we're approaching, maybe it's instead an animal cell, so there would be no cell wall. One of the key features of animal cells is that they have no cell wall. Well, we still would have a kind of structural support outside, but it's called the extracellular matrix. Outside of an animal cell, there is a network of different kinds of fibers which hold adjacent cells together. You have proteins like collagen fibers that are holding things together. You have a meshwork of glycoproteins and polysaccharides and proteoglycans and all kinds of things that are secreted by the cells, but also help hold adjacent cells to one another. The cytoskeleton that we talked about last time is actually anchored to the extracellular matrix through some of the membrane proteins that we're going to talk about in a little bit. So you can see here on this diagram that we have the cytoskeleton fibers, the microtubules on the inside, some of those proteins that make up the kind of skeletal network inside the cell. And those are physically attached to the extracellular matrix through some integrated proteins that are inside the membrane here. So you can see that each cell is attached to this extracellular matrix, and then you can imagine how many cells could be attached to one another in order to create tissue. Because if you pull on your skin, right, you know that your skin is made up of many different skin cells, but they don't just come right apart from each other, do they? It's your extracellular matrix that's holding that material together as a single tissue. So you want to make sure that all of your cells are anchored to something, and it's this extracellular matrix that makes it a cohesive fabric. On this diagram, you'll also see glycolipids and glycoproteins. You'll see these little kind of carbohydrate groups sticking out from the cell surface. A lot of these are involved in cell identification. So your red blood cells, for instance, have antigens sticking off of them, which represent your blood cell types. So a lot of these surface groups could be important in cell identification, letting your immune system know, oh, no, this cell is fine, this is supposed to be here, or hey, red flag, this cell is not supposed to be here, it doesn't have the proper authorization to be in here. And sometimes they come up in other forms of cell-to-cell communication as well, and we'll talk a little bit more about cell-to-cell communication as we go. Now, in plant cells, we talked about the plasmodesmata, the kind of tunnels through the plant cell wall to allow the movement of materials from one cell to another, and there are cell junctions that you find in animal cells as well. There are different types of cell junctions, so we have what are called desmosomes, which are strong junctions. You have these thick protein filaments running in between the cells that are really strongly creating almost like girders between these cells in order to fasten them together so that they won't come loose, right? If you had some muscle cells that you were trying to fasten together, you would want some good desmosomes in order to secure that connection so that they can't be pulled apart. You also have what are called tight junctions, and this is really good when you want to waterproof something, so think watertight in that case. So if you have a material that needs to be, you need to make sure that there's not going to be any kind of space in between cells for any fluids to leak out of. Your bladder, for instance, you wouldn't want any fluids leaking out into your body cavity, you would want tight junctions in between those cells in order to make sure that you're able to hold water, so to speak. And then you also have what are called gap junctions, which create little tunnels almost to exchange materials between adjacent cells. So there are different types of cell junctions. In Biology 110, I don't specifically ask you about the different types of junctions, so I don't have a slide with the different types on it, but know that there are cell junctions that allow the passage of materials. If, on the other hand, you are in my Bio 121 course, I will make a separate video about these 3 different junction types because I will ask you questions about these 3 different junction types. So that is a brief overview of the outside of cells. Let's start moving a little bit more towards the interior. Let's talk about the cell membrane. So as I said before, the cell membrane is a phospholipid bilayer. So here we have a phospholipid. Now, fortunately, I am not going to have to review everything we know about phospholipids because these are online lectures and pre-recorded, so you can go back and look at the lipids video if you want to remember anything about phospholipids. The important thing is that they have a hydrophobic region. These fatty acid tails are hydrophobic, and they have a hydrophilic region, this phosphate group head. That's attracted to water. The tails are repelled by water. So we call this an amphipathic molecule, and because it's amphipathic, when you mix them up in water, they will arrange themselves into these 2 layered sheets with all of the hydrophobic heads pointed toward an aqueous environment, a water-containing environment, and all of the tails pointed toward each other, toward the interior of this sheet. This creates a situation where we now have 2 separate aqueous compartments separated by a hydrophobic region. And this liquid oil membrane actually creates a barrier that prevents materials from moving from one side to the other. So if I were to mix, for example, a dye on one side of this membrane, that dye might not be able to move over to the other side of the membrane. So one might turn one color and the other might turn a different color. Or if I were to mix salt on one side of the membrane, that salt might have a hard time moving from one side of the membrane to the other side of the membrane. And this, very basically, is how cells maintain the difference between the inside of the cell and the outside of the cell. They have to maintain their own homeostasis, and they do it using this phospholipid bilayer. So the cell membrane is a phospholipid bilayer. Now, one thing that's a little bit difficult to wrap your head around is the fact that none of these phospholipids are locked in place. This is not a solid barrier. It is liquid. They are oily. They can freely move past each other. So in what way can a liquid be a barrier? It doesn't make a whole lot of sense to us in the macroscopic world, on the big scale, but in the microscopic world, an oily sheet like this can represent a really large barrier. To help you visualize what I'm talking about, take a look at these shade balls. Shade balls are plastic orbs that are used to cover drinking water reservoirs. Each one is just a single plastic ball, and it floats around on top of the reservoir, not physically attached to any of the other shade balls on the water's surface. And yet you can see that they naturally form themselves into these regular geometric, almost crystal-like patterns, fully covering the water surface, creating a membrane between the air above and the water below. And that liquid-like membrane is sturdy enough to prevent the passage of large molecules through it. So here, I'm using this clip with permission from the excellent YouTube channel Veritasium. You can see here Derek Muller being lowered onto this membrane of shade balls, and they are able to support his weight. They're able to prevent him from falling into the water below, even though they are not physically attached to each other. So they are able to freely move past each other. This is a liquid-like membrane, but it is still a substantial barrier to the passage of certain types of molecules. So that is the first function of the cell membrane, is that it is a barrier, but it's not just a barrier, it's actually a selectively permeable barrier. To be permeable means that things can permeate, they can pass through, they can move in and out. So things can get from one side of the cell membrane to the other. They can move into the cell or they can move out of the cell. Wastes can be excreted, and food and nutrients and ions can pass in, but selectively. It's selectively permeable. The cell can decide what to allow in and out. And the way that it makes these decisions is by using embedded proteins. So it's not just a phospholipid bilayer. We have these integral proteins kind of float around like icebergs in a phospholipid sea. Some of them behave like channels, little tunnels that allow materials through. And they can be very selective because proteins have specific shapes, so they can decide to only allow sodium ions in or only allow potassium ions out. Some of them are specifically shaped to only allow water molecules in and out. So this is the way that the phospholipid bilayer, the cell membrane, can select which molecules can permeate, is with these integral membrane proteins, with transport proteins in there as well. Now at this point you should start to realize we have these phospholipids, we got these glycoproteins, we got these glycolipids, we got these integral proteins. Another thing I haven't even had time to mention yet, but I'll go into it a little bit more in the next video, is that there's cholesterol mixed in with the phospholipids in this bilayer in order to regulate how fluid the membrane is. I've mentioned before that there are enzymes that hang around on these membranes, so they're doing a whole bunch of metabolic work. We got the extracellular matrix, we got the cytoskeleton fibers running along here. There's a whole bunch of different molecules, all of which are interfacing with this membrane. So it's not just the phospholipid bilayer. In fact, it's just a whole mesh, a whole network of different types of molecules, all of them working together in this kind of fluid mess here, a big tapestry, a mosaic, if you will. And in fact, that is the name that scientists have often given this model of the cell membrane. It's referred to as a fluid mosaic model of the cell membrane. All of these different molecules, all these different kinds of molecules working together to create a cohesive pattern, a cohesive whole that has a huge diversity of different kinds of functions. Let's take a look at a few of the different proteins that you might find embedded in the cell membrane. So I already talked about transport proteins, and some of these proteins just form little tunnels that allow molecules in and out, little back doors, little passageways that allow materials to come in and out of the cell. Some of them only allow flow in one direction, some allow flow in both directions, and some of them you can actually close up. You can decide, nope, we have just enough of what we need inside or outside right now, we're going to shut them off, so we can decide when we're going to allow flow and when we're not going to allow flow. And that can allow things to move in a passive way, what we call a passive way. It requires no energy to do that. They move the way they were going to anyway, but we allow them to, and then we can decide to stop them if we really want to. We can also allow active transport, which requires a little bit of energy. I mentioned when we talked about mitochondria briefly, and we'll talk about it a lot more in the future, this molecule here, ATP. ATP is the energy currency of the cell. It's what any enzyme, any protein needs in order to kickstart its function. Every time you spend an ATP, you can get a protein to do its job one time. That's the simple way to think about it. And this particular protein is a transport protein, so its job is going to be to pump. It's a protein pump molecules against their concentration gradient. What does that mean, against their concentration gradient? It's kind of like pumping them uphill. It's pumping molecules in a direction that they wouldn't naturally wish to go. Typically, things want to go from where they're most concentrated to where they're less concentrated. That is, they want to spread out. But using active transport, you can actually increase the concentration of something. You can pump something from where it's less concentrated to where it's more concentrated, but only if you use energy in the form of ATP to do so. So those are two different proteins, two different kinds of transport proteins, passive transport and active transport proteins. We also have some enzymes that you would find inside the cell membrane. So enzymes, of course, do metabolic work, they do cellular work, they speed up chemical reactions. You remember that from the proteins video that we did. So enzymes speed up chemical reactions. A lot of these enzymes are actually found on membranes. Mitochondria help do aerobic cellular respiration, right? Well, a big part of how they function is that they have a lot of surface area on their interior membranes, and those interior membranes are filled up with these enzymes that are ready to do their jobs. So a lot of these metabolic processes we're going to talk about, the enzymes that are catalyzing them, they're found on the membranes. So membranes are important as a kind of work table, a workshop space for enzymes to do their job. Another protein you might find on the exterior of a cell is a receptor protein. Receptor proteins have a little pocket on them that accepts a signaling molecule. That signaling molecule could be something like a hormone or a neurotransmitter. And once that molecule comes into contact with that pocket on the receptor protein, a signal is sent inside of the cell to help it change its behavior. So this is how, for example, two neurons could communicate with each other through a chemical synapse by sending signaling molecules from one to the other and interacting at the receptor level. So signaling molecules and receptor proteins. This is another kind of protein you would find inside cell membranes. I mentioned cell-to-cell recognition, so here's a glycoprotein and a receptor on another cell being used to identify that, yes, this is a cell that is allowed to be here. This is one of Professor Klein's, own cells. This is meant to be here. Don't worry, immune system. Don't start a response here. You could have cells that are joined together through protein connections. So that's another reason why you would have these intramembrane proteins. And then you also have proteins that will help attach the cytoskeleton on the interior of the cell to the extracellular matrix, which anchors tissues together. It anchors cells together in order to form these tissue sheets. So there are a lot of reasons, and this is only a few of them, that you would have these membrane proteins all around. So cell membranes, these are things that you would find in every single kind of cell. This is not just animal cells. All cells have a cell membrane, and all of them would have these kind of proteins somewhere in their structure. Their basic structure is the phospholipid bilayer that we discussed in the lipids video, but they have way more complexity to them than that. They have all of these different molecules working together, and that's why I refer to it as a fluid mosaic. Sometimes it's easy to get lost in a lot of the language we use to describe cell membranes because there's a lot of terminology. Fluid mosaic model, that is the model. That's our way of conceiving of the cell membrane. It talks about all of the different molecules working together. Even though it's a liquid membrane, it still forms a selectively permeable barrier. Selectively permeable means that things can move from one side to the other, but only what the cell wants to move from one side to the other when it wants it to move there, and it makes those decisions by utilizing proteins that are able to control that kind of movement. We'll talk about that in the next video. Phospholipid bilayer refers to only the most basic structure of the cell membrane without all of the extra proteins and other things included as well. So while it would be correct to say that the cell membrane is a phospholipid bilayer, you wouldn't just point to it and say phospholipid bilayer. You would call it the cell membrane. Membranes of this type are generally referred to as plasma membranes. The big one that surrounds the entire cell is referred to as the cell membrane, but all of the ones on the interior, like the membrane that makes up the endoplasmic reticulum, you would just say that's a plasma membrane. You would never call an interior membrane a cell membrane, if that makes sense. Alright, I think that's enough for a video. In the next video, I will talk about transport across membranes.